So yeah, thanks to Andy for the invitation, and thanks for your persistence in getting this meeting to actually happen. So I think that's really good. I was invited to this 4 years ago, so it's great to actually be here. So, I mean, to start, everybody knows this. Antibodies amongst the most used research tools. Researchers purchase antibodies from commercial manufacturers, and some percentage of antibodies, of commercial antibodies, do not perform as advertised. But what that means is really mostly anecdotal, and I'll have some data on that later. It's been a problem for a long time. And the issues are well understood in this room. Validation of commercial antibodies is variable. There's no standardized criteria or SOPs, no real comprehensive or independent framework, batch variations, variable validation methods. And this is sort of part of a driver of some issues with reproducibility in science. So how we got into this, not because of antibodies, we got into this because of an interest in my lab in neurodegenerative and neurological disease. So we've had a long-standing interest in ALS, amyotrophic lateral sclerosis. It's a disease you do not want to get, devastating neurodegenerative disease. And in 2011, there was this discovery of a gene called C9orf72 as the major ALS disease gene. And I mean, this is far and away, there's more ALS caused by a mutation in this gene than all the other genes combined. In the Finnish population, 21% of everyone who has ALS, period, has a mutation in C9orf72. And so it encodes a protein of unknown function, and the protein contained a protein module called a D-domain. This was a module that my lab had identified and had shown was involved in membrane trafficking. So strong supposition that this protein was involved in membrane trafficking. We wanted to understand the link between membrane trafficking and this neurological disease. So what a cell biologist, a trained cell biologist like myself does, is if you want to understand what a protein does, you want to understand where it's localized. So we started by going back to the literature, and we found a paper that said C9 is localized to the nucleus, and then we found one that said it was in the endosomes, then we found one that said it was in the Golgi and the autophagosomes, we saw it was cytosol, then we hacked it. Okay, it's possible that the protein is localized everywhere in the cell, or it's possible that the lack of consensus on the localization stemmed from antibodies. Okay, so this is all we're doing. I'm not particularly interested in antibody validation. My postdoc, Carl LaFlamme, and I are interested in C9orf72. So we sit down one day and we go, well, there's lots of antibodies out there. We should be able to go through the commercial antibody repertoire and find some antibodies that are specific so that we can just go ahead and do the cell biology on this neurological disease gene. So we decided to use CRISPR-Cas9 to knock out C9 and some cell lines, purchase every C9 antibody we could get from every commercial supplier, and screen through them. So there's Carl. That's what happens when you work on C9. I've burned a lot of people in my lab trying to work on this gene. So here's what we did. Carl made some knockouts. He made a heterozygous knockout where we knocked out C9 on one allele, or we made two complete knockouts where we knocked down C9 on both alleles. This is somewhat important because there's some concern in the literature about whether C9 is a hypomorphic loss of function or a dominant negative gain of function, but that doesn't matter for this talk. So we made this sort of hypomorphic knockout, or we made two complete knockouts, two different lines, and we purchased every antibody we could purchase, okay? And we ran lysates where we had the parental line and the isogenic controls, or the knockout lines and the isogenic control, I guess being the wild type, the parental. We ran a bunch of blots. We stained them with Ponzo so that you could see that the bands or the protein lanes are evenly loaded. And we started doing Western blots. And here's an antibody. That's one's not bad. See? It's a band here, sort of goes away with the knockout. This one, not so good. That one, not so good. Here's a beautiful one. Here's a beautiful one. Go through them all if you want. It's all published. Here's one that doesn't look so good, okay? So we went through all of this kind of data. And we'll focus on these three for now. So here's one where you can see a beautiful band at 45, and it's kind of half loss and then complete loss in the knockout. Same thing with this one. This one, okay, there's a band at the right size, and you can imagine it would be easy to get misled. Now, I'm going to deliberately not talk about the names of the companies as I go through. So I'm not going... I'm not going to tell you the name of that company. I'm not going to mention the name of that company. But maybe you can guess. All right. So it's funny until you recognize this. So these two monoclonal antibodies, one from Abcam, one from GeneTex, that were optimal for immunoblot, another one from GeneTex that was optimal for immunofluorescence, and IP. None of those antibodies had ever been used in a published manuscript. The antibody from Santa, Mr. Tom Cruise, had been used in 32 publications that had been cited greater than 6,000 times, okay? That antibody, I can guarantee you, I don't care what the validation approach, does not recognize C9orf72 in any application, okay? So it's a problem. And we don't really feel it's as much a science problem as we feel it's a business problem. So it's expensive to test antibodies, okay? It's not cost effective. The people in this room know this way better than I do. I mean, you know, scientists say manufacturers should test, rightfully so. Granting agencies and journals want testing, but they don't enforce it. Manufacturers believe at least some of the onus should be on the scientists, and that's absolutely true. Scientists say we don't have the money, we don't know how, you know, it's not required by the journals and the granting agencies, so why should I, okay? And all told, that leads to some estimates that maybe $1 billion are wasted a year on bad antibodies. But worse, well, worse, $1 billion is pretty bad. But maybe worse is, you know, a lot of non-reproducibility that costs downstream, okay? So we came up with a solution. It's not the solution. I had it labeled as the solution, but it's just our solution. So we created a concept for antibody characterization or testing that we call YCharOS. So YCharOS stands for Antibody Characterization through Open Science, okay? So we envision YCharOS kind of as similar to like a bond rating agency. It's like Moody's or Standard & Poor's, or a product rating agency, Consumer Reports, or a product safety organization like, you know, highway safety, things like this, okay? YCharOS is an incorporated company, okay? It's, yeah, we could make some profit, I suppose. I can tell you one person who's not making any money off of YCharOS. It currently has one part-time employee, who's the CEO, who's Chayton Rainin. The company's incorporated in Toronto. But all of the work, 100% of the science has been done in my academic lab at McGill University. All of the money that goes to the YCharOS initiative is either granted directly to my lab, so it goes to McGill University through a research grant, or there are research contracts that sometimes go to YCharOS, and then through YCharOS are granted to my lab or contracted to my lab. Okay. And all credit here to Elle Edwards, someone who I think many of you know in this audience. Elle's quite an incredible person, and you know, we were doing the antibody validation. Carl and I were doing the science, but the idea is all Elle's, okay? Elle's the one that really put this initiative together, okay? So this is the ecosystem. And so we created a trusted open science ecosystem, and I want to emphasize a few things here. So on the left are the granting agencies, the funders. On the right are our trusted partners, and these are the antibody manufacturers, and many of them are represented in this room. And I just want to state very publicly that this YCharOS creation, this YCharOS idea does not work without these partners, okay? So the way it works is that we talk to the funders, and the funders, let's take an example like the Michael J. Fox Foundation, Parkinson's Disease Research. You know, a huge interest in trying to validate Parkinson's disease antibodies for their scientists, right, for the Parkinson's researchers to use. The NIH, this is a program on Alzheimer's disease. Safari, this is a program on autism disease research. These funders want validated reagents for their investigators. So they get together with their scientists, the experts in the field, and the scientists come up with a list of proteins, targets, that they want antibody validation for, because their scientists are screwing around with the antibodies and they'd like to know where we're at. We present those targets to the partners. And the partners, so the targets are not selected by us, they're selected by the funders. The partners select the antibodies and send them to us. Us meaning my lab / the YCharOS initiative in my lab. We do the scientific work, and what that involves is we make the knockout cell line, or from one of our partners, Abcam, and is one of our partners that provides us with knockout cell lines. And so we either obtain knockout cell lines or we create the knockout cell lines as needed. And that's based on various approaches to trying to find cell lines, human cell lines that express the protein of interest. And then, so we take the wild type and the knockout and we do the immunoblots, we do the immunoprecipitation, and we do the immunofluorescence. And all of that data is then presented to the partners in monthly meetings of our board, which we call the crack. And so many of the people around this room are on crack. It's an old joke. It used to work in the 90s. It doesn't work quite so well anymore. And all of that data, after internal review by the partners, gets immediately put onto an open science server called Zenodo. We also are now publishing some of those reports. That's only to try to increase the visibility. That's the only reason we try to publish those reports. Okay? So this is a public good. There is nothing in this. So for-profit, not-for-profit. YCharOS is owned by a charity called the Agora Trust Charity. There's no investors. There's no conflicts of interest in any way, shape, or form. All of this data is immediately released to the public. I know I'm spending a lot of my time working on this in my laboratory at the expense, I would almost say, of my basic research on the cell biology of neurological disease. But we are doing it because we feel it's an important public good. Okay, so then what we do is once we have the data, we put them together into these small reports, antibody reports, where we publish the standard operating procedure that had been agreed upon by everybody on the crack board. Okay, so we use those agreed upon procedures. We present the data in its rawest form possible. We put it onto Zenodo. You can find it at Zenodo or the YCharOS website, YCharOS website. And so far, and this number keeps changing. So 75 antibody reports, 730 antibodies tested. That number keeps going up every day. Okay. So the data I'm about to show you might be a bit old in some cases, more antibodies than others. Okay. So now that we've got that number of antibodies validated, we can begin to draw some conclusions, right? So some good news. Taking a data set of 75 targets, mostly neuroscience-related targets, selected not by us, but by funders. And in many cases, these are what we would call dark targets. These are the NIH program, for instance, that nominated all the Alzheimer's disease targets. These are targets that they've selected through research that aren't really, well established as Alzheimer's targets. Many of the ALS targets are ALS genes that are kind of new to the ALS field. So these are what we would call dark targets. So these aren't well-studied proteins. So I think this is a pretty good representative data set. So the good news is that how many of those targets are covered by at least one antibody that we say works? And we try not to use that word too much, but for this kind of data, you have to draw some conclusion. So in Western blot, we could find an antibody that recognized the target in 86% of the targets, 89% for IP, 61% for immunofluorescence. Not too bad. And if we looked at the renewables, monoclonals or recombinant renewables, the numbers were still pretty darn good, 77, 75, and 54%. We could look at the performance of all antibodies. And by the way, this is a paper that's currently under review. Reasonable performance of all antibody classes. So this is the success of antibodies for Western blot, for immunoprecipitation IF. Again, the recombinants appear to outperform the polyclonals and the monoclonals a little bit. Same thing for IP and IF. Okay, so that's kind of the good news is that it looks like just with the existing antibodies, assuming our 75 targets are reflective of the 20,000 genes in the human genome, it looks pretty good. You guys have generated antibodies that are pretty good. Now, the sort of not so good news, a substantial percentage of antibodies do not really perform to recommendation, okay, so according to our approaches. So here's the antibodies for Western blot that were recommended. So if you go to the catalog of the manufacturers, these antibodies were recommended for use in Western blot. 578 antibodies. 79% of them in our approach were specific for Western blot. They worked. They immunodetected in Western blot. The definitions here are a little tricky. 21% did not. That's pretty reasonable. Of the antibodies that were not recommended for Western blot, half of them still worked. So there's almost an underselling of the products. And that even becomes more clear with the IP. Here, most of the antibodies are not, there's just no, it's not that they're not recommended for IP, there's just no recommendation. We test them all in all procedures independent of what happens in any other procedure. And 37% of those things that aren't even recommended, or just there's no recommendation, all work. So again, the manufacturers are probably underselling the quality of their products, right? I think the real problem comes here, and this is, I think, if I go to my standard cell biology meetings where I go and talk about RABs or talk about neurological disease, this is something that comes up all the time, okay? Even though of the antibodies that were recommended for immunofluorescence by the manufacturers, in our knockout approach, under our procedures using our SOPs agreed upon by the manufacturers, 39% of those antibodies actually work, 61% do not. That's a bit of a problem. And the problem gets worse with this, is that there's sort of improper use of antibodies. So we went using CITAB and went back into the literature and said, okay, of all the antibodies that we've now done our validation approach on, we could find 2010 papers that used one of those antibodies in Western blot. 31% of those papers used an antibody that didn't pass our validation approach. 69% used an antibody that did pass our validation approach. So, you know, we're not the purveyors of everything right here, but at least as we look at it, there's some issues there. So I'm going to close now. It's at 18 minutes. Told you, right on time. So 2 more slides. YCharOS and its partners, I think, are helping to change Icarus. I came here calling it YCharOS. I'm the only one that calls it YCharOS. Everyone else calls it Icarus. And I'm one of the sort of founders of the thing. So I'm now trying to work on Icarus. We're changing the landscape. So here's, I think, a really nice example of how Icarus has helped change the landscape with our partners. So these are antibodies against a gene called PARK8, Parkin, one of the major Parkinson's disease genes. And everybody in the field uses this antibody and this antibody. And I can confirm that because, you know, I talked to the Michael J. Fox Foundation. I talked to my colleague Ted Fawn, who's one of the world's experts on Parkinson's disease, and we do a little bit of work. And look, not bad. Parkin's a 50 kilodalton protein. There it is. There it is. Okay, we talked yesterday about antibodies. Is that antibody and that antibody the same antibody? Yep, it sure is. Okay, 2 different manufacturers. But look, it works. But would you want to really spend a lot of time using that antibody? So we presented this data back to our partners, and one of our partners came back and said, look, here's a business opportunity for us. They went out and made 2 recombinant antibodies. Look at that. That's, thank you, Matt and Thermo. And these antibodies work brilliantly, and my understanding is they're doing quite well. And the Michael J. Fox Foundation is now telling their investigators, if you're going to do Western blot, stop using those ones. I'm sorry, you got to use these ones. And in fact, our manufacturing partners have started to either remove or modify their recommendations on their antibodies based on the feedback they're getting from us. One last slide before I conclude. To bring it full circle back to one of the reasons I got into this, ALS. Okay? So one of our early funders were the ALS Societies of Canada, of the United States, and the Motor Neuron Disease Association, as they call it here in the UK. It's not ALS, it's the motor neuron disease. So it's called the motor neuron disease. So we validated antibodies against 27 ALS disease genes that are linked through classic genetics to ALS. We did Western blot IPIF. The check mark means it passed validation. The X means it didn't, and the dash means we didn't get a conclusive answer. So what these are lysates of cells, motor neurons, cortical neurons, dopaminergic neurons, oligos, astrocytes, and microglia, all derived from iPSCs. This is not a trivial experiment, believe me. As well as fetal astrocytes, fetal microglia, and human macrophages. And we used our knockout-validated Western blot antibodies, and what we find is that at least for 1, 2, 3, 4, 5 ALS disease genes, they're not expressed in motor neurons at all. They're expressed in microglia. And this is a new emerging concept in the field. And you can only do that by having these kinds of really powerful reagents. So I'll close by thanking Carl, who really drove the thing. Al Edwards, who is he's our guru, or Svengala, or whatever they call him. Chayton Rainin, our CO, and all the folks in the lab that do the work. Thank you very much.